Hi everyone. Today I'll be describing the molecular interactions that a cell uses to migrate through an extracellular matrix. Before jumping into this process, I'll be giving you some background info on the most important components of this process. Actin, integrin, myosin, and fibronectin are all important components that aid for cell movement through an extracellular matrix. The extracellular matrix, or ECM for short, is a highly organized structure with the capacity to direct cell migration through the tendency to follow matrix fibers. Actin is a motor protein that contributes to muscle contraction, vesicle and organelle transport, cell mobility, and cytokinesis. In this case, we'll be focusing on how actin aids in cell movement. Actin filaments, usually in association with myosin, are responsible for many types of cell movements. Myosin is another molecular motor that converts chemical energy in the form of ATP to mechanical energy, thus generating force and movement. Fibronectin is an extracellular matrix component that, through binding integrin proteins on the cell surface, acts as a key player of the communication between the intra and extracellular environment, thus controlling cell behavior. Integrins are the principal receptors used by animal cells to bind to the extracellular matrix. Now that we know the main components needed for cell migration, we can now describe how cells are able to move through the matrix. Cells generate actin proteins at the leading edge of the cell through polymerization, causing the leading edge to protrude forward along an ECM fiber. Integrin then binds to the ECM fiber, and clutch proteins anchor integrin to actin filaments. Integrin is able to bind to the receptors of the cell surface thanks to the help of fibronectin. Integrin activates the signaling protein, and that signaling protein then activates myosin. Myosin is able to slide actin filaments at the back of the cell, causing contractions. This overall causes the generation of contractions, causing further protrusion at the front of the cell, which creates movement, which allows the cell to move to whatever location it needs to be at.